remember saying to Erica, my wife, that I think I'm going to get a job.
Dr. Ross Levine is the Chief Scientific Officer at Memorial Sloan Kettering and one of the world's leading physician scientists studying cancer.
All of us have moments when things don't go well, and it's hard to keep going. And I think that your job as a mentor is to help people at those moments.
As a fellow while working in his mentor's lab, one day as he was about to head off on a family vacation, he got a very interesting result. He canceled the vacation and stayed home over the holidays, poring over the DNA sequencing results. Those sequences revealed the JAK2 mutation, a single genetic typo that drives a whole class of blood cancers and which he co-discovered. 6 years later, there was an FDA-approved drug.
Focus on what you believe based on what you see, what patients need that we don't have today. And if you focus on that, you can be part of delivering it.
We explore what that discovery taught him about the gap between finding a target and truly hitting it, and why he believes the biggest breakthroughs in cancers are still buried in data we haven't yet analyzed.
Welcome to Stanford Department of Medicine's Inside Look at the Future of Medicine.
Welcome to the Future of Medicine and welcome to Stanford.
Thanks so much. It's great to be here.
So we've just come from a really fantastic medical grand rounds, where I think you gave a really inspiring talk. Certainly I found it inspiring, and I know our trainees, our residence fellows will be very inspired by your career trajectory as a physician scientist. But before we dive into your career, I want to take you back 21 years ago, I think right now. You were a fellow working in the lab, and you made a really significant discovery. Along with several other groups, you co-discovered the JAK2 mutation, which we know is a key mutation that causes a type of chronic blood cancer called MPN, or myeloproliferative neoplasms. Following the discovery of that mutation, within 6 years, there was an FDA-approved therapy. So one of the most rapid translations from discovery to FDA approval in the history of hematology oncology. So maybe you can take us back to, what was it like in the laboratory at that time? Was there an aha moment all around the discovery? And then, what has it been like for you over the last 20-something years, just watching the impact of that discovery?
It's a really good question. I remember most of what I was doing at the time was just doing lots of analysis of Sanger sequencing done at scale of all of these patients. And I remember that we were doing lots of sequencing. We'd see things that were interesting. We weren't sure if they were worth pursuing. And I remember that we were doing both MPNs and we were doing some leukemia sequencing. And I remember that we had two different plates of samples. One was 84 AML samples, and the last 11 and then a control were MPN, and the next plate was all MPNs. And I remember we were sequencing through the JAK2 gene, and I was just scrolling sequence by sequence, and the first 84 AML samples were negative, and all of a sudden there was this run of mutations. And I remember being excited, and Gary and I talking and saying the most important thing we need to do is to confirm this, because if we can repeat it, then we know we have something. And I remember that it was over a holiday, and I canceled a vacation. Encouraged my wife and infant son to go without me to visit family, and I stayed back, and I did the sequencing. And I remember it came back, and I was just sitting at the kitchen table in my house in Boston, townhouse in Boston, and saw it. And I knew at that moment that a lot was going to change, that we were going to have a diagnostic that was going to be incredibly useful almost right away, and that therapeutics were going to get developed. Whether they work or not, I didn't know. But I was pretty aware that this was this pretty amazing moment. And I remember saying to Erica, my wife, that I think I'm going to get a job. I also know that the hardest thing about science is that discoveries are so hard, and they're so unpredictable, and they're so ephemeral. And I think the hardest thing for a physician scientist or PhD scientists, trainees, is you can go through years where you don't make a discovery, and you don't know when it's going to happen or what it's going to be. But when it does change your sort of narrative and your journey. And it was that moment both for me and for the field more broadly.
Yeah. So of course it was a critical discovery, right? Really laid the foundation of how we diagnose these these cancers, and how we treat them. So it's so foundational to the field. But maybe there are people listening to the podcast who are not hematologists like you and I. So maybe you can share and explain a little bit about the JAK2 gene, what it does, and what happens, or why is this mutation, which is really a very small mutation, a single amino acid, as you outlined in your medical grand rounds presentation, one amino acid change. Why is that so problematic? Why does this cause so much trouble for this gene? Maybe you can explain a little bit about that for our non-hematology audience.
Well, I mean, we have 518 kinases in the human genome that place phosphorylation marks on other proteins that are substrates. 90 of them are tyrosine kinases that phosphorylate tyrosine residues, and JAK2 on first principles is one of those 90. And in fact, our project was to sequence all 90 because we were not so thoughtful to figure out which one was worth sequencing. In retrospect, I think it was obvious. But JAK2 has a very special role. It and the other three JAKs, JAK1, JAK3, and TYK2, all play a critical role in really being the kinases that mediate signals from cytokines outside the cell to the nucleus. And they are the catalytic subunit that makes cytokines go. So for example, erythropoietin given to to stimulate red blood cell development, either exogenously or endogenously, EPO binds its receptor and then it activates JAK2. In fact, JAK2 is the only kinase in the human or mammalian system that can transmit the signal from EPO. So if you had to pick one kinase that would be important in red cell apparent proliferation, it's the one, it's obvious. And so the way I think of it is that JAK2 is one of many, but it has some very unique roles. And in retrospect, it was probably the single most important kinase in myeloid blood specification and proliferation. And what probably is occurring in these chronic blood leukemias, it's co-opting a normal lineage sort of factor that promotes their specification and proliferation. And like in many times, cancer is incredibly adept at sort of co-opting the mechanisms that the normal tissue it emanates from and using them. It just then, as you said, this small mutation, through mechanisms I'd argue that we don't fully understand, it's like taking a gene that normally can be in the on or off switch, and now it's stuck in the on switch. How and why? I think people think they know. I'm not sure we actually do know what the mutation does. Does it activate the enzymatic activity? Does it block the domain it's in? Does it facilitate dimerization? I actually think these are all plausible hypotheses that are not proven. I'd argue actually the person who's probably done the most work to study that in the world is Chris Garcia, who's a faculty member here, who's published the cryo-EM structure of JAK1 2 years ago. So I think there's a lot to know, but the important thing is that that one amino acid change, as you said, is tiny but mighty, and it takes this gene that normally is a regulatable signal and it becomes an unremitting signal.
Right. So incredible to think that people have been working on this for 2 decades, and as you outlined, we don't really fully understand the nuances of this, which I think is a perfect segue to therapeutics, which was obviously the other. Not only was this so important diagnostically to be able to definitively diagnose people with polycythemia vera or any one of these chronic blood cancers, but ultimately the therapeutic impact has been huge. And as I mentioned, we had the FDA approval of ruxolitinib 6 years later, 2011. We now have 4 JAK2 inhibitors approved. And although these, of course, have been incredibly helpful for patients, definitely have had a positive impact, they have not been the whole story, right? And they have, as you mentioned in your medical grand rounds, they are non-curative therapies. And so maybe this relates to part of the nuance of the mutation and how it affects the function of the kinase. So what have we learned from all that 20 years? What have we learned from the therapeutics, where they've helped, where they haven't helped? What is left to do? How will we do better? And maybe you can also talk a little bit about your own work in that area, which you also mentioned in the medical grand rounds presentation today.
I mean, I think what we've learned is that the current drugs, first of all, they have no ability to inhibit the mutation versus the wild type gene. So at some level, if you push their dose, they're going to inhibit a lot of the wild type function, as we said, because it's important for red blood cell development and white blood cell, and to some degree for platelet development. You're going to get some on-target toxicity, and we've seen that. But what we've learned, I think, and this is just a candid perspective, is that partial inhibition of the pathway, including a lot, but not all of the inhibition of the mutant signaling, say 40 or 50% of its signaling, is good enough to shrink spleens, reduce the aberrant inflammatory sequelae, and probably improve survival, but it's not going to lead to disease regressions. Like, that's the state of play. Where we were in 2011, it's where we are in April of 2026. And so that tells me that we need better medicines. And so we've spent a lot of time trying to ask some questions. We did a lot of work asking whether the pathway was reactivated in the patients who don't regress. We asked, and the answer is yes. We took mouse models and cell lines and asked in cells that are not responding to these current drugs, if you, for example, knock down JAK2 or reverse the mutation, do they regress? They do. And so all the data we had in 10 years of work in the lab, which I think in many cases was trying to build an argument for a second attack on JAK2, honestly, was that everything we had was that this pathway's on and it's dependent. We just aren't getting there with the drugs. And you know, a lot of what we did, honestly, was that we just couldn't convince people to do it. And I think, you know, what happened ultimately was that we were able to launch our own effort, and we can talk about that. But then a lot of other companies, I think as they've learned more about the structure, and probably most importantly that the chemistry has gotten more tractable, that we can target other domains like the pseudokinase domain, and we can do better and better job between cryo-EM and AlphaFold and chemical biology. I just think things became chemically possible that weren't possible. And so we've now seen a number of approaches. This includes targeting the domain where the mutation is in, the JAK2, which is actually a pseudokinase with partial enzymatic activity. So you can engage that domain and there may be mutant selectivity there. The drugs that have been developed are not mutant selective in their binding, but maybe in their activity. As I said on medical grand rounds, we focused on binding the inactive conformation, the type 2 conformation, whereas the current drugs are type 1. And I still think there will be yet additional strategies that may emanate, blocking dimerization, other mechanisms. But I think what I'm excited about is that we're now seeing a new wave of innovation. Yeah. And these fields are funny. Like, how much of that is because the science wasn't there? Some of it. I think some of it is that, honestly, people got caught up in immuno-oncology and sort of stopped doing it. And then, you know, I think you sort of reinvigorate. And then I think, to be candid and maybe a little pejorative, you know, the drug development field is very much a field of few leaders and many followers. And what you find is once you get one or two people starting to do it, like, you know, some of the JH2 work, some of our work, then now you're gonna have 10 companies doing it. And it's an unfortunate reality that we see versus 10 distinct innovative strategies. But the good news for patients is I think there's renewed sort of substantive innovative effort to bring new therapeutics. And I think what I'm most excited about for our patients is that between the JAK2 strategies, the targeting calreticulin, which you've worked on, and others, maybe going after some other exciting targets in these diseases, I think we're going to see a whole new wave of therapeutics that are in and will be in the clinic, and it'll be fun to see how those play out alone or together.
Yeah.
And I'm here for it.
Yeah. No, I think it's an incredibly exciting time. We just had this, you know, series in Blood, the new wave of targeted therapeutics, and I think obviously JAK2 is a big part of that. The molecule that you've developed, we have other companies, and I can say as a physician, having these multiple clinical trials to offer to our patients now is really fantastic, and patients benefit from having, you know, people working in the space. But it is really interesting to remember that belief that this was the right target and it was the drugs that were deficient was really very important in us in getting there.
And I would let others judge. I by no means ever believe that I have this, I'm not saying I was the only one who was banging that drum, but I think I was one of the louder people banging that drum and spent a lot of time trying to convince people. I'm like, there's something to do here. And like at the time, I think it was hard to get people excited. And now I think, you know, and you just have to, like, I think as a physician studying a disease, you have to say, all right, that's the journey. Sometimes it takes you time to both get the science there and to get the community there. But if the need's there, you gotta, like, it needs champions like us.
Yeah. Totally. So we're going to zoom out a little bit from JAK2 now. I think one thing that I found really intriguing, just looking back at your original paper of the discovery of JAK2, and you mentioned that you sequenced the kinome, right, which was, I guess, 19 tyrosine kinases. And at that time, that was a huge thing, the ability to be able to sequence, all those kinases all at once. And, fast forward 20 years, and now, genomics is such a part of what we do. Like in the clinic, we have targeted sequencing, we have whole exome, we're talking about whole genome. Genomics is such a part of our lives now in the clinic and also, obviously, in the research arena. So, I mean, what's your opinion on sort of how genomics has served us over the last 20 years? Has it, where has it really exceeded expectations? Where, has it sort of, our hopes have not been met? And where do you sort of see the future going in that regard, in the whole sort of genomics field?
It's a great question, and I would say that it's one that I've evolved my own thinking on, so I'll just take you on my own perspective, right or wrong. I think I had a pretty deterministic approach to this when I started my career. I was like, there are drivers, we're going to find them, and then we should drug them. And JAK2 is a great example of that. And I started with that idea that, like, I cannot imagine that sequencing many more patients will find things that really we go, oh my God. And I was sort of like, all right, like we'll sequence 1,000 MPN patients and do all the genes and we'll have the answer. And I think that where I've been, I think, pleasantly surprised to be and happy to be wrong has been that in the both germline space more broadly across human disease and in the somatic space in cancer, every time we achieve a level of scale of patients, we find things we didn't know about before, whether it's new genes, new patterns, new things. And as you begin to link data and numbers of patients at great number with many other features, whether it's their clinical outcome or their response to a drug or their immunological parameters, we find things we didn't know about before. And so I actually think, this is just an opinion, that the germline field is massively lapped the somatic cancer genetics field in this regard. Thanks to things like UK Biobank and Regeneron's effort with Geisinger and some of the others, the germline genomic data is in the tens, if not greater, hundreds of millions. And what they've shown over and over again is even by finding protective alleles or pathways, you find things you didn't know about. And that also teaches you that with that scale, you find biology that you didn't know about before. Cancer genetics isn't there. It's just not. Like we have like, hundreds of thousands of patients maybe, if we aggregated maybe 1 million or 2. And I was very much of the ilk a couple of years ago that it didn't make sense to ask whether we should scale. And I think we have to. Like I actually believe that we're gonna find things. Now it may be unexpected things. It may be like protective secondary alleles, whether they're germline or somatic, that might be good drug targets. It may be pathways and linkages. And I just think like we have to just say that the data, once we achieve it at scale and with all the other kinds of data you can acquire, and of course the analytics, we're gonna find things we didn't know about.
Wow.
And so I'm all in on it. I think you have to and I think MSK has made the decision in the cancer space that generating as much data from as many patients that we have our arms around is gonna be our greatest way to innovate and achieve impact. And the way we approach it, probably different than almost other places, I can't speak for Stanford in this, is it's an institutional effort. There's not a breast cancer effort or a leukemia effort where you sequence every advanced cancer patient, and then that data is aggregated that night and available to the community. We just don't know who's going to figure out what to do. We don't think it should be one it should be a group. It should be democratized and available. And we are very eager to find other partners who want to do that so we can achieve collective scale that we can't do individually.
I have to say, a little surprised. It was not surprising that would be the answer you would give. I thought you would say, oh, well, sort of most of the discovery is done, and our real deficiency is that for most of these recurrently mutated genes, we don't have therapies or effective ways to target them.
That's also true.
I guess they're not mutually exclusive, but I guess your point is that you think there's still a lot of discovery to happen in cancer genomics. And I guess how do you think about that for the field? It seems to be saying that all cancer centers, right, should be really leading this, and then obviously we should have data sharing. Or do you think there needs to be more sort of concerted, organized efforts, like we've had things, right, like in terms of the AML consortium and others? What do you, or is it all of the above? How should these questions be?
It's a really good question. I'll start with my concern in this space, and maybe we can build from there. My concern in this space is that most of the cancer centers have largely ceded the sequencing to third-party providers, Caris and Tempus, and name your favorite group. That's great for the patient in front of you because they get the result they need. These companies are good. I have great confidence in their skills. But like, who owns the data and who's innovating with it? The answer is if for-profit entities are doing it and it's somewhat opaque to the, I'm very worried about that. I'm worried that, patients don't want us to have their data primarily in proprietary companies when it's being paid for and reimbursed as part of their care. I just don't think that's what patients want. Patients want us to have that data available, both whether locally or institutionally. The problem with that is that the infrastructure you need to build to do that without those companies is not clinically, financially feasible. So an institution's got to say, this we think will fuel discovery and we're going to invest in it. We'll try and get some of this reimbursed. And very few places, like we got a $100 million transformative gift 12 years ago from the Kravis family. Like you can't do that without that kind of scale. But our perspective is that we've gotten much more back in discoveries and in grants. And also like we can go to companies and be like, we can find the patients with relevant biomarkers and we don't have to ask somebody else. Like we can look it up today. And so it's a decision. It's interesting. We're always on this existential journey of whether it's the right way to do it. But my belief is that academic institutions have to be very thoughtful that they are the place of data generation. And the innovation is going to come from having access to as much of that data as possible. So anything that limits your ability to see it is bad. And the second is like, I always think back to like, what do the patients want us to do? Right. And so I've never met a patient who said to me like, I'd rather not have my data sequenced at MSK, it'd be rather at Caris. Like if it gives them, of course they'll do it. But if you ask them what they want is they want people to discover things with their data. Right. And not, like, of course there are economic realities, anyway.
Yeah, so I think, of course, obviously genomics is a big part of, discovery and so critically important, but now we have so much more, right?
Yeah.
We have CRISPR, gene editing, we have all the signal cell technologies, we can get very high resolution information from a single cancer cell in a patient. We can get this really, really detailed information, and of course we have AI, which we're just beginning with, right? We're just on the tip of the iceberg. So, what are the questions that we can ask now that we couldn't ask when you started? Where is all this going to bring us in the future in sort of in the cancer space? How do you see this playing out?
Ooh, it's a hard one. I mean, I would say in some ways that the greatest thing that these tools let us do is look at more features over time. And we can correlate those to clinically meaningful outcomes, whether it's response or resistance to a drug, whether it's progression or metastasis, or whether it's transformation from MPN to AML. There are aspects of all of those phenomena I just described that are not understandable with the current understanding of cancer genomics. Like none of them are fully solved. Like I don't, for example, in our world of leukemia, I don't think we understand mechanistically p53 mutant leukemia and what else is going on. So there's things missing. And so I say first and foremost that we need these tools to understand the questions that we don't fully understand. The second is like, I think these technologies, when you get into more powerful things like spatial, I still think we're in the early days. They're pretty descriptive, to be candid, in my view. They're important, they're interesting, but they're descriptive. And I don't think we fully understand what to do with them yet, but that shouldn't prevent us from doing it on lots of patients and seeing if we learn things. And so I'm excited to begin to see that data generated. But if you ask me, do I think today is the day to do spatial on 100,000 patients? I'm not sure I'm there yet. I think we're getting closer to it every day. And then on the AI machine learning aspect, I think like, maybe this is simplistic and wrong, but maybe it's not. Like, I think that there's a lot of things that we see in medicine that, like, we have this innate sense that there's something causal or correlative that we can't put our mind on. Everything from, we know for specific molecular subtypes of leukemia, when we look under the microscope, they look different than other molecular subtypes, like the nucleolus of certain, and like you ask a pathologist, they'll be like, yeah, of course. And it's been very hard to quantify and to lock those things down. And there's a lot that the human mind can't even process. Right. And so my view is that that's the exciting part of this, is that sometimes it's going to be genomics. Sometimes it's just going to be measuring a CBC over time. Right. And sometimes you can do either of those things, and they're both measuring the same thing. And then we can ask the interesting question of, do you need the fancy technology, or is there a more clinically like low-tech thing that measures the same? I think that's the journey we're on now. And then the more interesting thing is, like, when you start thinking about multimodal data. I tell the story that in 2012, we published, I thought, one of our most important papers, which was the genomics of AML and 500 patients, right? It was a large-scale study, 18 genes, 500 patients. Right? Just for perspective, you think about that now. It's like one day's work. But it was important. And for about 5 years after that, every doc at MSK was a leukemia doc, and many docs around the country would email me with like their cases. And they'd be like, Ross, I have a case with mutation X. And I'd be like, well, based on the algorithm, this is what I think, this is the prognosis. Right. I mean, they could look it up themselves, but I had it memorized. And I remember when Ellie Papamanuel and Peter Campbell's larger AML patient sample came out, I think it was about 1,530 patients from Germany with heart donor and 110 or 111 genes. What they did, Moritz Gerzong and Ellie and that team did, was they created a website where they could like plug in the genetics and the age and the white count, and it would spit out. And literally it went from calling me and like what I figured out to like, people would send me an email and I'd be like, plug it into this. And like, that's the era we're entering. It's not that these individual things aren't meaningful, but there are much greater precision when you amalgamate multiple data sets. So I'm intrigued by it, and it'll be fun and interesting.
Very exciting time.
Like the JAK2 mutation, bear with me for 1 minute, is an ET, where people can live for 50 years, and it's in MF high-risk patients that live for 5. Clearly, the JAK2 is not the biomarker that distinguishes those. It has to be other factors. Things, and are those measured with genomics? Or I think that's the journey we're on,
are they
right?
Measured with hematopathology, AI?
And our patients, like, they just want to know what journey they're on, and can we give them, precision, and then can we design next generation trials for the higher risk patients versus not the low risk.
Totally agree. There's so much opportunity there. There's so much we don't understand about why some people progress and others don't. So want to shift gears a little bit now just into your role as leader and mentor, which I think in my opinion, your mentoring part of your career is really one of the most impressive parts of your career. When I look around the country and I look at people who've trained with you, who are leading their own labs, leadership roles, thriving in academia. So that's really been a big part of your career, I think. So, but if we summarize your current roles, right? You're the Chief Scientific Officer at Memorial Sloan Kettering. You're the principal investigator of your own laboratory. I think up until a very short time ago, you still saw patients in the clinic. You've founded a company to really focus on targeting the JAK2 V617F mutation. So pretty busy number of roles. So first of all, how do you manage to do all the things that you do? And secondly, why is it important for you to have these roles? And particularly, why is it important, after 20 years, you're still in academia, you're still mentoring people, you're still doing science. What is it about those things that keep you here as a physician scientist?
That's a lot of really good questions. I'll start with the last first. I've been blessed with so far a great career and with great colleagues, and I've had great opportunities in academia. I've had opportunities in industry that have come up and have talked about that both for myself and talked to lots of friends who've gone on to do great things. I have lots of colleagues from my peer group that are leaders in pharma, whether it's Jay Bradner at Amgen or Mark Dawson at Roche or Levi Garraway. I have these incredible role models and friends that have gone on to do great impact. I think for me, a lot of it is that the difference between academia and industry, and this is overly simplistic, but I believe true, is that pharma, although it develops people, is prioritizing the project. And ultimately, a really talented person will be moved from one important project to another because the project can get killed. And usually talented people survive, but sometimes they don't, and they move on to others. Academia is much more about betting on the person.
Yeah.
And to me, what I've always enjoyed about all the parts of what I've done, whether it's running a lab, whether it's having leadership roles, my main job is to find really talented people, to figure out with them how to them get focused on something, figure out what they need, and then do two things, only two things. One is to get out of their damn way and just let them be. And then the second is all of us have moments when things don't go well. 10 experiments in a row don't work, and then you read that someone's actually done the thing you were trying to do, to personal challenges, and it's hard to keep going. And I think that your job as a mentor is to help people at those moments.
Right.
And I think I felt like I've benefited greatly from helping people at those moments and seeing them thrive. And the reason I took on these leadership jobs was mostly because you get to do it at bigger scale. You get to think about individual PIs or even larger efforts and say, who are the great, what I think a lot about at MSK, especially in the current climate, is that especially in the current funding Headwinds, how do we ensure that the best and brightest, who I think are going to be the leaders for the next 15 years, how do we recruit, retain, and keep and resource those people? What I don't want to see happen in the current climate is for those people to reduce the scope of their science. That's my job. My job is to resource those people and to let them follow the data and their curiosity where they go. And that's super important to me.
So I think that is so important, and I think you and I both care so deeply about physician scientists. But I guess if we dive into that a little bit deeper, there are a lot of challenges, right? We know that of all the physicians who graduate in the United States, maybe less than 5% end up becoming physician scientists, and that number is dwindling. We know there, as you mentioned, the NIH pay lines are tight. There's a lot of pressures, clinical productivity pressures. I'm sure these pressures existed when you were training, but it feels the sustainability of the physician scientist path is really something that we have to really pay close attention to. So I guess just first of all, I assume the answer is if you were to choose the path you took, you would choose it again. But how do we make sure that path is there for the next generation, and for maybe for physician scientists who are early in their career right now? What kind of words of encouragement do you have for them at this time? It's
a great question. I would choose it again, but I think that it's good to take a step back and ask what are both the headwinds and the tailwinds. I think that the way I view it is there have always been historical headwinds, right? You're asking people to train in two different disciplines, science and in medicine. You're asking them to do that into their mid-30s. You're asking them to bet on that working out for them versus maybe taking a path of, for example, a clinical path where maybe there isn't the same sort of risk-reward ratio. And you're asking them in many cases to defer, both personal and life choices till they have some stability. So you're asking people to go into their mid-30s or later without certainty on their path. That's a hard thing for people to do, and probably has gotten harder in the last 20 years, just based on how we are as a society when we think about those things. But that's been around a long time. I think, though, that what has gotten harder is that you layer on a couple of things. You layer on, as you said, the funding landscape. It's harder for them to get their first grant and second grant than ever before. The second thing is that the number of jobs and academic institutions aren't hiring, so it's harder to get a job. And then the thing which I think we all talk about but nobody has been able to solve is it's getting harder to publish your papers. Every paper takes 5 years to do and 2 years to get published. And so I worry about all those macro factors. I think most of them are not unique to physician scientists, but they're exacerbated by a group of people who have to do this dual training and emanate from it in their 30s. And people are appropriately saying, why am I doing that? I think that my opinion is twofold. One is that we need physician scientists. We need them in academia. We need them doing what you and I do. We also need them in industry. We need people who do what we do and then go to industry. The budget for drug development and discovery in industry dwarfs academic medicine and the NIH budget. If we don't have the best and brightest using those resources, that's not good for society either. So we need a pipeline in both. And so the answer is we need it. And so I think that you have to think about it two ways. One is, are there macro barriers that have increased in size that we can attack? Like, how do we think about the funding landscape or publishing or all of that? And are those unique to physician scientists or not? But the second is that I think we need to develop more bespoke strategies for physician scientists from the minute they start. Like, if we don't create more of a pipeline with some sort of safety in it, it's just people are going to be like, why? Yeah. And so I'm worried. And I think that no institution yet I've seen has done it systematically. And what I hear and challenge for all of us is the basic science community says, well, is it really that different that in our world where 1 of 50 PhDs gets an academic job? And the answer is, I don't know if it is or it isn't. I don't think all the challenges are unique, but what I see is it's a disproportionate reduction. And as somebody who's an MD without a PhD, I see that group's the most, like, there are almost like no new MD only folks running labs. I think that's unfortunate, because I do think there are people that go through their training, may not have thought when they were 20 and they wanted to be a scientist, and they become sort of like the clinical experience shapes them and drives them to science. And if we don't find a path to support those people to do it, I think we need to do it. The hard part, I think, and this is what I say all the time, is that when you go into this path, you have to realize it may not work out. And that's a hard thing for high achievers to realize. Like, you want to go into a path knowing that if you do everything right, it's going to be okay. And it's hard, right? Like, you may not.
Yeah, there's no guarantee of success.
But you're a clinician. And like, there are many things you can do with the skills you do that might be different. And that's good. Like, I believe that the training we have allows you to achieve impact in many other ways. I think, getting back to your mentorship point, one thing I learned from my mentor and yours, Gary Gilliland, was that it's not fashioning people in your own image that's the mark of a great mentor. It's allowing people to achieve what they want to achieve and helping them navigate that. Some people, if you're a great mentor, are going to follow a path similar to yours because they'll see that you enjoy it, there's impact. But some people are going to say, I don't want that. I want something different. And my question is, well, how do I help you do that? Not, it's a mark of failure that you're going down this other path.
Yeah, no, so I think these are all great points. I mean, I think at the institutional level, at the individual PI level, we need to see the value of physician scientists, right, and find ways through resources or how we organize ourselves to really make that a feasible career. But I guess on the other side, so if you're the trainee at this point in time, do you have any thoughts or guidance for them about, it seems to me, and even looking at your career, I think it's a key example of that, sort of like early decisions or early exposures in your career that were really foundational to your career. You mentioned, I think you spent a year in medical school where you're at HHMI fellowship, and then obviously having a great project to work on when you were a fellow. Are there things that trainees should be doing, early decisions they make, early training they make? Are those the same things that you should have been doing, like when you were at that point, or are they different now? Because obviously it's a different world now. We have AI, we have all these tools, we have large-scale data. Do you have any kind of advice of sort of how you sort of try to help position yourself best for success if you were training?
Yeah, it's a good question. I'd say two things, but there's so many more you can sort of articulate. One is that for me, I look back and I say, what was the most important thing? It was having an incredible group of mentors and peers that helped me navigate it. It wasn't just the lab PIs I worked for. It was the person who encouraged me to go into lab before all of that. It was a pathologist named Bob Kerman. It was some of my clinical mentors who backed me when I wanted to be a scientist and not a clinician. Like, I had a lot of people. You want to find those people. And I always say, like, you find great mentors, you find great folks who are sounding boards, and you know, like sometimes you don't follow their advice, but often you do, and that's an art of knowing when to follow advice, but go get that advice. I always tell folks that the best thing a trainee can do is expand their mentoring network. Like go to a meeting, I call it stickiness. Go to a meeting, meet someone amazing, and then email them after and be like, thank you so much. I loved the brief interaction. I would love for the opportunity if I have in a moment. Half of the people might not write back, but half will. And the ones who will be on your list. And if you say to them a year later, you go to the hematology meeting, can I talk to you for 20 minutes over coffee? The good ones will do it.
Right.
And they'll do it because you show initiative. And
Right.
I think trainees should show that. That's the thing that distinguishes, I think, the people that are going to do great more than it's this ability to sort of show initiative and find make your own mentoring environment versus asking someone or go get it.
Right. It's a drive.
Yeah,
I guess.
and it's finding those people. That's the first thing. The second thing is I do think that it's important. When I was a postdoc in Gary's lab, every other person in the lab was making mouse models. It was the de rigueur at the time. And Gary, I remember, went in the lab when I was starting, and he's like, we got to get into genomics. And he got 4 of us in a room, and we all agreed to go to the meetings at the Broad. And the other 3 people within 2 months left, and they went back because the mouse models were
straightforward.
It was what we did well.
Yeah, yeah.
And I was I think willing to do and help us figure out what to do next.
Yeah.
And I always tell trainees, and whether that's AI or single cell, I don't know, I think it's great to have some of that idea. Don't work on the thing that 5 other people before you did.
Right.
If you find the mentor who's got will help you inculcate the idea of what's next.
Right.
And will support you. Gary had my back. He's like I believe in this and I believe in you with this.
Right.
I didn't ever have to worry about at some point I was going to bankrupt the lab because half the money was going into sequencing, and Gary's he got up at a lab meeting and he showed it, and he's like, yeah, I just believe in it, and it may bankrupt us, but I think it's probably going to work, and he's the guy to do it. And I remember he was in front of the whole lab, and in the end it paid off. We made discoveries, and obviously it actually led to a whole next wave for the lab. But I think that.
But it's you getting out of your comfort zone.
Yeah.
And it's the trainees getting out of the comfort zone. So sort of don't do the thing that you know how to do well and keep doing it over and over again, right? You and this trainee period is such a unique period where you have to learn the skill set, right, to launch your lab, right? So to use that time to sort of get out of your comfort zone and do hard things and things that you don't know how to do.
And the other thing, and I think this may be a little generational, is I think that it's easy in training to do what's expected and then to leave and go do the rest of your life. And there's nothing wrong with that.
Yeah.
But the trainees that I think always make a mark find a way to think about something, whether it's talking to someone about a question or they're thinking about what they did in their day, and they're getting thoughts about what's next. You want to follow those impulses and ideas as much as possible.
Yep.
You may not be able to act on them in full during your training, but you should be doing that.
Yeah, no, totally. So I think we're coming close towards the end now, and I put together some rapid fire questions for you. So maybe we'll fire these at you. Before we conclude. So here's the first one. What's a scientific question you desperately want answered but you don't have the tools to address yet? Tough question.
I really want to understand the individual cell transcriptional and epigenetic signatures of rare mutant populations, which I think is not achievable today.
I see. And not achievable because you can't find the cells, or because?
I don't know how to measure mutations and other features with incredible granularity. I think people either do single cell and don't get accurate mutations, or we get some of the single cell mutation data on the other. So I think once we can begin to do that with great and get multimodal, which includes clonality in those single cell analyses, for what I do, it's going to be exciting. That's more of a technical need, but important. We're going to go after it.
I see. Got it. Okay. So I can imagine there'll be a lot of implications for that for these rare cells. In terms of AML relapse, the cell that comes back.
We
10
can be
years
broader
later.
and be I was saying earlier today in a one-on-one meeting, we've put all of our AML work. We've moved it from the leukemias we've historically studied, which are these epigenetic regulators, all the P53 and EVI1 mutant AML. Because what I want to work on now is the leukemias that kill people that we don't understand, that need treatment. So if you ask me like the bigger question, that's the one that keep like
this highly
these awful leukemias.
that are resistant to everything.
That's the one that,
yeah. And then, okay, so here's another one. If you could go back and give your intern, one piece of advice, what would it be?
Whew. Intern Ross.
Yeah. This is before you even got into the lab. Well, you had a little bit of exposure
I had a little
as
bit of lab.
med student, right? But
I don't know. I think at that stage, I probably would have encouraged myself to try to like learn more from others, even more. Like I was so focused on just surviving, and I don't think I realized how much amazing intellectual and like firepower there was around me. And I look back, and it was mostly seeing amazing people under duress, and I think there was a lot to learn, and a lot of people I wish I still had more opportunity to engage with now that were remarkable, but I only just saw them in fleeting moments.
I see. So you've talked a lot about mentorship, and you've mentioned your mentor, Dr. Gary Gilliland, many times. So what is the best advice you've ever received from a mentor, from Gary or from someone else in your career?
Well, I do think back when I was a senior postdoc in his lab and things were going well, and I wasn't really worried about transitioning to an independent career. And he said to me, we were in his office, and he's like, I'm thrilled for you to stay here and continue to collaboratively work on stuff and put my name on the end of your papers. But he's like, it's time for you to go out and do it your own damn self. And that was like, he's like, you're ready. He's like, go do it. He's like, you're not going to know what it's like. It's going to be all these challenges. It's going to be frightening. He's like, just go do it. And I don't know, I know that's one piece of advice, but it was funny because I was not someone who's like agitating about I was having a blast. Like I was,
you would have been okay.
Like, I was publishing great data. It was good. You know, like I was really happy. And he's like, it's time for you to go and do it.
Okay, so he probably saw it before you.
He did.
That's
Absolutely.
Awesome. Yeah. Okay, we're book, scientific or otherwise, that's influenced your career or thinking.
I think Outliers, Malcolm Gladwell's book.
Okay.
For those of you who don't know, that's the book about 10,000 hours and the idea that you don't get good at something until you put a lot of time. I really believe that. I think like that's the whole physician scientist thing. You have to do the hours in both. And I think that book is, for me, incredibly insightful about what allows people to be successful, and that combination of experience and then having sort of drive is really important.
Awesome. And then, we've talked mostly about your career and your science, but of course I know you have many other things in your life. You run marathons, you have a great family, but what's something that people would be surprised to learn about you? Or is there anything that, you're also a person who is always sharing and talking, and so I don't know, what's surprising about Ross Levine that we don't know?
I would say that, at my core, for someone who I think often comes off as a gregarious extrovert, like I have a lot of moments where I'm not, and over time more of them, I would say. When I started in this, I really did enjoy always sort of being out there and in the community and all of that, and I do. But over time, I think I've become more of like someone when I'm not doing these things, I need to do the exact opposite.
So you like your quiet time.
I like my quiet time, I like my runs with my friends, I like just sitting with the family. Like I've become much more like, and whether that's pandemic driven or not, but it definitely, I'm more of a homebody than I used to be.
Not
and
quite
I think
an introvert though.
No, but it's funny, I think I don't necessarily look for these opportunities in front of big crowds. Like they're part of what I do. I enjoy them in the moment, but I don't seek them out. And if anything, I would prefer to let others have those opportunities, and for me to like stay home and watch my favorite sports team with my family or friends.
Awesome. So this has been such a great conversation, Ross, and I know your Medical Grand Rounds presentation has been so meaningful, especially for our residents and fellows. And so I'm going to end with the final question. To maybe any medical student or resident who's listening to this, and they're just listening to this conversation we're having, and they're looking at your career, and they're thinking, this is what I want to do. I want to have that kind of impact, bench to bedside, really impactful for patients. So what is one thing that you want that person who's listening to know?
Focus on what you believe based on what you see, what patients need that we don't have today. And if you focus on that, you can be part of delivering it.
Awesome. Well, that is a perfect way to end, Ross, and thank you so much again
for
coming to Stanford and for participating in the Future of Medicine podcast.
Thanks so much.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.